You always make my slides look rather inferior, I have to say, David.
Well, that's because, you know, usually I get a lot of help from other people.
And I see you've certainly got a Twitter account now.
I've got a Twitter account. You know, I was actually forced into doing that by my students. They said I was very old-fashioned, and so they've counselled me through this. And I wouldn't exactly say that I'm an old hand, but look, it's a bit of fun, you know.
Yeah, certainly. It's a good way to reach people.
Yep.
Well, I think by my clock, it's 9:05. So given we've got an hour slot, I think we'll get going. Others may join us as we're going. But good morning to all of those, or afternoon to all those joining us today, and welcome to today's talk. A quick note just to say, I hope everyone is staying well and safe during this very difficult and challenging times. I know it's been a fair bit of change for everyone. My name is Richard Mackenzie. I'm based in London, Roehampton University, and I'll be hosting today's talk. My area of research, for those that are interested, generally sits within the same theme that we'll be talking about today, which is insulin resistance and glucose metabolism. Now, it's a very, very special thanks to Professor David James, who's our speaker today from the University of Sydney. Now, Professor James really needs no introductions, but I want to give him one anyway, so his work certainly deserves it. I first came across some of his work during my own PhD. His early work made significant contribution to our understanding of insulin's action, the actions of insulin, and particularly in muscle. And in the 1980s, for example, he published a series of landmark articles in Nature describing and characterizing the fundamental nutrient transporter that is GLUT4, its responsiveness to insulin. He holds a great number of honors and awards. It's difficult to know which ones to really bring up. I know he's blushing as I'm talking now, but these include Glaxo Wellcome Medical for medical research, and more recently in 2016, he was awarded, which was new to me, the New Zealand, the Premier Prize, Excellence in Medical Biological Sciences. And he also holds an honorary doctorate at the University of Copenhagen, which was awarded in 2019. Today's talk will focus on more of his recent work titled Systems Analysis of Exercise Biology, a talk I've heard a little bit before, but I'm very much looking forward to hearing more today. So without any further delay, I'll pass you over to Professor James, but just a quick note to say that if any of you've got any questions, most of you should be, all of you should be muted. If you've got any questions, you can email me or use the function on Zoom to ask me some questions. And if we have time towards the end, I'll pass these on to Professor James and we can discuss these. So please, Professor James, over to you.
Thank you, Richard. Everybody, you can hear me okay, Richard? That's okay? Yeah?
Yes.
All right, great. Well, thank you so much, Richard, for the kind introduction, and thanks everybody for joining in this morning from London. It's unusual times. I hope that this can bring, you know, at least for an hour or so, a break from the torrid and boredom of lockdown. So I thought what I'd do is just give you an introduction to the work that we've been doing over the last probably decade now on the use of protein phosphorylation as a tool to understand complex phenotypes. Before I start, let me just introduce some of the players. So pretty much everything that I'll talk about today is a collaboration with the University of Copenhagen. And there's a photo of some of the key people there. There's Eric Richter and Bente Keynes and Jørn Wojciechowski. They're the main partners in crime in Copenhagen. And the latter part of the work is a collaboration with Pfizer Pharmaceuticals. and there's Christian Permuller behind there. And then the young lady standing next to me in the front is a very clever and talented PhD student in my lab, Elise Needham, who's really been driving a lot of this work. And then some of the other work I'll talk about was done by postdocs who've now left the lab, Ben Parker and Nolan Hoffman. Okay, so we decided to use protein phosphorylation as a tool to probe function for a number of reasons. As is displayed here on this slide, one of the main reasons is that I would argue that almost every single, at least key biological process involves a change in protein phosphorylation at some level as a key regulatory driver. But secondly, and perhaps more importantly, unlike other parameters such as gene expression and things of that kind, protein phosphorylation is a much more dynamic process. You can see changes in this process within milliseconds in cells. And finally, because of recent advances, particularly in mass spectrometry, it's conceivable to measure changes in the phosphoproteome of cells or tissues really at quite a global and quantitative level. So for these reasons, we have been focusing on this for a number of years. And I should just emphasize at the outset that, there's a great opportunity here for not only discovery, but also, making key insights into diseases and other biological processes. So we now believe that in humans that the phosphoproteome is vast. I mean, this is the prediction. There's probably many more sites than that. And what's interesting is if we look at the total number of reported sites, which is, some hundreds of thousands, really we only understand the function of a tiny proportion of this total network. And therein lies one opportunity. Now, of course, each of these sites is, the phosphorylation of these is catalyzed by a protein kinase. And, there's always this desire when you are studying protein phosphorylation to map that back to a regulatory kinase. And again, there's tremendous opportunity here. So despite the fact that there are more than 500 protein kinases in the human genome, we've only been able to map, again, a tiny proportion of this total population of phosphosites to a particular kinase in the genome. And in fact, for many kinases in the human genome, we actually know very, very little of who they phosphorylate and what their function is. And, we believe this is actually a very big oversight. Now, this is just a sort of a way that we're trying to display this phenomenon. So here you see all of the protein kinases in the human genome. And what Elise has done here is she has assigned them a substrate, and that's based on the size. And I'll just run that. And so she's now collapsed that down into this network. And you can see that based on the size and the color, they're the ones that we know a lot about their substrates. And you can really see that the majority of protein phosphorylation sites that many of us think about and focus on really sort of condense down into just a handful of key protein kinases. And I'm sure these are kinases all of us have heard about, things like ERK, AKT, mTOR, and so on and so forth. So really, what do all these other kinases do? That's one question. So what I would like to focus on today in the context of protein phosphorylation then is exercise. Now, again, I'm sure everyone in the audience is well aware exercise has tremendous health benefits. And in fact, in many diseases that extend even just beyond muscle itself, you know, even things like cognitive function. Exercise is well known to benefit cognitive function. So our thinking was that if we could look at all the signaling pathways that are triggered in skeletal muscle upon exercise, that would begin to give us a much deeper handle on all of the beneficial effects of exercise that, you know, I think many of us would like to understand, so that we could then potentially develop therapeutics that mimic some of those events. Now, again, I won't belabor all of the work that we've done here, but the first study, which is summarised here, and again, this was where we began with Copenhagen, Eric and colleagues, where they, as they want to do, took 4 young healthy Danes, submitted them to a fairly hefty but short-term bout of bicycle exercise, and took muscle biopsies from each one of them before and after, immediately after the exercise, sent those to us, and Nolan and Ben in the lab performed a workflow whereby they isolated phosphopeptides and then quantified those using a mass spectrometry. Now, in this particular case, it was a labeling strategy called TMT, but we use various approaches to quantify these phosphopeptides. And I hope you can see this. This is a volcano plot, which sort of displays those phosphorylation sites that we quantified as being significantly changed in response to exercise. And, you know, strikingly, we identified in this study more than 1,000 regulated sites. So now that's, of course, an underrepresentation because this is not, you know, completely comprehensive, but it's certainly much more comprehensive than I think any of us had seen before. And so herein for us was an opportunity to understand, you know, what are all these sites telling us about what's going on in muscle during exercise, and which of these are encoding the beneficial effects of exercise in the aftermath of an acute bout of exercise. Now, many of you know that one of the key kinases that is regulated in muscle during exercise is the stress kinase, AMP kinase. And indeed, we observed activation of AMP kinase in these data. And here is an analysis of the data where using machine learning, where you effectively just scan through the data, look for known AMPK phosphorylation sites, and use that information to interrogate all of the data to ask what other sites are behaving in the same manner as those known AMPK sites. And shown at the tip of this so-called Pareto front are the highly predicted AMPK sites, and they're displayed here in this blow-up. And you can see in red there all the known AMPK sites, such as acyl-CoA carboxylase and so on and so forth. But in blue, we were able to identify in this study at least 6 completely novel AMPK substrates. And indeed, we've gone on to study some of those, and they have revealed novel biology that is regulated by exercise. And I'll come back and talk about that now. So in the next study, and this appeared last year in the EMBO journal, and this was performed by another postdoc in the lab, Maren Nelson, in collaboration with Ben and Nolan and others. Here, what we were interested in doing was trying to take that sort of exhaustive list of phosphosites and narrow it down into a sort of higher confidence compendium of the key regulatory nodes that occur in muscle with exercise. And to do that, we use different paradigms of exercise in mice or rats, and then used all of that data together to come up with a convergent list of the most robust exercise regulated phosphorylation sites. And I'll show you that on the next slide. But again, this was the sort of the summary synopsis of that paper. And here you can see that one of the proteins that we identified in this screen is a protein called STIM1. And this is a very interesting protein because it is at the core of regulating store-operated calcium entry into cells. And we performed a fairly exhaustive series of studies to analyze the phosphorylation of STIM1, showed that it indeed is phosphorylated by AMPK, and in so doing, AMPK plays a key regulatory role in regulating store-operated calcium entry into exercising muscle. Again, a completely sort of novel arm of AMPK biology. Now, on the next slide is just a sort of a summary. You can see there a human, rat, mouse of all of the sites that were consistent amongst all of these different paradigms of exercise. And what's interesting, you can see there in orange, some of the proteins, LMOD2, here's our STIM1 and another one here, were actually phosphorylated not just on one site, but in this case, 3 sites, others on 2 sites, which sort of gives you a sense again of the of how what a priority exercise has designated these different proteins in terms of presumably regulating their function. Now, many of these proteins, we have no idea what they're doing in exercise, and each one of them, I would argue, presents an opportunity for future investigation. And I often sort of jokingly say at this point, I mean, we focused on STIM1, one of the proteins on this list. That was a pretty focused effort from 2 postdocs in the lab for at least 1.5 years. So if you wanted to sort of work your way through this list, it would be a pretty decent amount of work for quite a few people for many years. Now, if we just bioinformatically go through this list and ask, well, what sort of functions are encoded there, you can see that there are a lot of cytoskeletal regulatory proteins. There are some transcription regulatory events. Cellular proteostasis is high on the agenda, and signal transduction. So many protein kinases and some serine-threonine phosphatases are on this list. And again, I won't belabor the list other than to say that I think it's a rich resource for whomever would wish to dip into this list and work up some of these targets. So if we take our list then of exercise regulated phosphocytes and then use some of the tools that are available to ask, well, which protein kinases are implicated then as regulating those sites. Here are is the major list that Elise and Peng Yi have come up with. And on this list are some of the well-known exercise-regulated kinases like cyclic A kinase, CAM kinase, and AMP kinase, as I've already discussed. But there are a number of other kinases which are on this list, again, never before implicated in exercise. Now, if we now also, because, you know, protein kinases are often regulated by being phosphorylated themselves. So in addition to just looking at the sort of known kinase substrate relationships, what Elise has done here is ask, well, what kinases are also phosphorylated in this data? And you can see that there are many, as I described on the last slide. And again, many of these are quite novel, and we were intrigued. Here's one here. It's actually called BIKE, BMP inducible kinase, which is rather fortuitous that it would be implicated in bicycle exercise. So one wonders if there could be such a kinase for different kinds of exercise, like walk or run, etc. So now, so then in the next series of studies that we published, we were, as I said at the outset, we're very interested in using this compendium as a way of sort of figuring out which parts of this network are encoding the beneficial effects of exercise. And I won't belabor this study today because it's published. It was published in Cell Reports last year. But just to give you the concept, because we're rather excited about this. So it's very difficult to sort of go into mice or obviously humans and start genetically modifying various molecules. So what we had the idea of doing was to use small molecule drugs in cell lines, in this case L6 myotubes, and then use the drugs to stimulate these cells and then perform phosphoproteomic analysis on each of those cells, either with one drug alone or multiple drugs in combination, and now use the phosphoproteome to map back onto the exercise phosphoproteome so that we could begin to disentangle this network. And again, as I said, I won't belabor this, but it was a very exciting outcome, because one of the things that we observed, so here, for example, was the exercise phosphoproteome. And using this approach, what Elise observed, for example, was that the drugs, two different drugs in this case, mimicked one part of this network, which was phosphorylation of a protein that regulates the expression of another protein called SERPIN-E1. And many of you have probably heard about this. It's a soluble secretory protein that actually has rather deleterious biological consequences. It regulates EMT and other various pathologies. Now, this was very interesting because when this molecule is phosphorylated, it's said to downregulate the expression of SERPIN-E1. And, you know, for many of us who are interested in studying the beneficial effects of exercise, particularly in looking for secretory factors that are generated by the exercising muscle that, you know, travel to other sites. And I think that's displayed here in this. I actually can't see that part of my slide. I hope you can. Can you see that, Richard?
Yeah, it is working. Yeah, working really well.
It's buried under all the pictures of all of you, but I think I know what it says. So it's an exercising muscle, I hope, and hopefully, oh, let me go back. That was not right. And it's hopefully you can see that there are molecules circulating through the blood. Now, typically when people have done this sort of an approach, they're looking for molecules whose secretion is increased with exercise. But in this case, what Elise observed by combining phosphoproteomics with secretomics was that, in fact, the expression and release of this molecule was actually decreased with exercise. And so this is a very, I think, innovative way of thinking about this, in that exercise may not only increase the production of beneficial factors, but it may also inhibit the production of deleterious factors. So, and that's what she observed in this particular study. Okay, so what I want to talk about for the remainder of tonight's lecture is a completely new study that we are in the middle of doing right now in collaboration with, again, Copenhagen and also Pfizer Pharmaceuticals. Now, as many of you know, and I've alluded to this, one of the beneficial effects of exercise is to increase whole body insulin sensitivity, okay? and many people have published on this since the early 80s. It's been shown in animal models and humans, and even in type 2 diabetic humans, that exercise training has quite a profound effect to improve whole body insulin sensitivity, okay? So again, we were interested in using similar approaches to what I've just described to see if we could disentangle these beneficial effects of a single acute bout of exercise on insulin sensitivity in humans. Now, the approach that we took is actually summarized there for you on that slide. This is a model system that the Richter and Wojciechowski lab have used over a number of years, and it's the one-legged exercise model. So they bring subjects in and they require them to sit in a chair, and one leg has to exercise as vigorously as possible for 2 hours, whilst the other leg remains at rest. Okay, so you've now got this sort of internal control. You can biopsy the exercising leg and the rested leg in parallel and do direct comparisons within the same individual for biochemical changes. Now, I should also say, and you know, these sorts of studies sort of send shivers up and down my spine. They catheterize the artery and vein of both legs and sample so that they can actually measure the AV difference across the muscle bulk of the leg. And by measuring various metabolites in those samples, you can get a sense of things like glucose utilization, either in the rested versus the exercised leg. Now, to make matters even worse, they don't stop there. They exercise the subjects. Oh, I think this is summarized. Oh, sorry, just let me briefly go back a second. I always like to talk about GLUT4 for obvious reasons, as Richard said, in whenever I get an opportunity. So glucose uptake into tissues is regulated in the manner in which is displayed in this slide. So here what you're looking at is fluorescently labeled GLUT4. In this case, it's expressed in an adipocyte, but I would hasten to imagine that exactly the same paradigm would hold true in skeletal muscle. And what you're looking at here is, so if we just wait until the video begins, which is now, so all those bright dots are GLUT4 vesicles, and you can see bright flashes, okay, at various points. And those bright flashes correspond to the fusion of a GLUT4 vesicle with the plasma membrane of the cell. So that's what we're looking at here, is that this is a technique called total internal reflection fluorescence microscopy, which focuses on the underside of the plasma membrane. And you can see that with insulin addition, this process is accelerated. So that underpins insulin action in muscle and fat. Okay, so here's the study design. So we do the exercise in one leg. They then rest for 4 hours, take a biopsy from both legs, then perform a 2-hour hyperinsulinemic euglycemic clamp. Now, I'm sorry, without knowing the audience, and this is where these sorts of formats are really difficult. I often get a sense of how the audience is going at this point just by looking at them. So maybe half of you are already asleep and I don't even know about it. But a hyperinsulinemic euglycemic clamp, for those of you who don't know, is considered to be one of the gold standards for looking at insulin action, certainly in humans. So what you do is you infuse insulin at a constant rate, and then in parallel, you sample the blood glucose and infuse glucose in addition to insulin in order to prevent the blood glucose from falling, which is what it would do if you were not to infuse glucose. And the amount of glucose that you need to infuse during this period is, of course, proportional to the degree of insulin sensitivity of that individual. Okay? So they perform this technique, but of course, because they've got AV catheters, they can actually look at glucose utilization either in the rested leg versus the exercise leg with insulin after exercise. So it's a very elegant protocol. Now, here is the data. So let's first look at glucose uptake across the muscle of either the rested leg versus the exercise leg, and that's color-coded either black or blue. And here are is the average. I think there were, I forget, 7 subjects in each group, something like that. Sorry, these are paired studies, right? Okay. And so here you see the well-described potentiation of insulin sensitivity. So you can see that the blue curve is much higher than the black curve. And this is the uptake during the euglycemic hyperinsulinemic clamp. And you can see that the leg that is previously exercised takes up much more glucose than the previously rested leg. So this is absolutely consistent with what we have come to know and learn. Now, I just want to show you something, though, that we were quite struck by. And this is a representation of the average data of all of these subjects. And I think, you know, this is sort of the way in which many of us have approached biology over the last 30 years. You know, you often take 2 groups, diabetics versus controls. You go and get 10 or 20 of each. You do something, and then you take the mean and the standard error of each data set and you ask, is there statistical differences? And off you go and publish your paper. But you know, when we looked at the individual data, we were struck by the incredible variance in the responses that were observed in these data, which was completely hidden when you took the average responses, okay? So here is one subject, for example. So this is subject 5. So you can see there is subject 5's resting response in the rested leg, and there's the exercise leg. So indeed, within a subject, you see the potentiation. But here's another subject, also potentiated, nowhere near as much, but this subject's potentiated response is actually not even as great as the rested response in subject 5, okay? So this actually really caught our attention. Now, if you think that maybe we just don't know how to do a glucose clamp, and of course we did worry about that at the time, I just happened to be going back and reading some very old papers. I don't know if any of you ever do this, and I don't often myself, but in fact, this has been one of the benefits, at least for me, of being in lockdown. I've started to do this, and this was one old paper, 1981. First author, Colterman. This was from the Jerry Olewski lab. Very famous paper, where they described for the first time, again, euglycemic clamps at different insulin doses, the phenotype of either lean control individuals, of obese insulin-resistant individuals, or individuals with type 2 diabetes. And here you can clearly see that insulin resistance results in a rightward shift of the dose-response curve of glucose disposal. And for type 2 diabetes, this response is almost non-existent, okay? So this is the kind of defect that we're all interested in curing, okay? Now, in contrast to what most papers do, these guys actually showed the individual data. And here's the individual data. And again, strikingly, what you can see is there is an extraordinary degree of variance in responses within individuals, even in the lean controls. I mean, some of these lean controls are actually worse in terms of their insulin sensitivity than the so-called obese insulin-resistant subjects, okay? So this is something that we thought about and wondered if we could actually take advantage of in our study. So, you know, as I said, most people get alarmed at this and they think, oh my God, it's so variable, we'll never learn anything. There's too much variance in the data. But rather, what we decided to do is maybe this is real biological variance. And if we could utilize this within the individuals in our study and ask, do we see a corresponding variance in protein phosphorylation in muscle, which we could then use to filter out noise within the data, which would enable us to sort of focus in on the key phosphorylation sites that are truly driving the phenotype of interest. In this case, both insulin-stimulated glucose uptake and its potentiation with exercise. So that was the paradigm of this study. Okay, so again, we did our usual, where we did phosphoproteomic analysis on the muscle biopsies, and here, as this is displayed by Elise, there are a number of comparisons that we can look at here. We can ask, what's the effect of insulin versus basal in the rested leg? We can ask, you know, what's the effect of exercise versus insulin, and so on and so forth. So there are a number of comparisons that we could look at. But for the purpose of tonight, or sorry, this morning, I just want to share with you a comparison of insulin rest versus insulin after exercise. So can we start to find phosphorylation sites that are predictive of that potentiation? Okay, so that was our objective. So again, if we look at glucose uptake at these two different times where we took the biopsies in the subjects, you can see that there's lots of variability between subjects, as I've already discussed. But in general, and certainly true for each subject, there is a potentiation of exercise. Okay. So the first thing we did was we asked which phosphorylation sites that are regulated correlate with glucose uptake within individuals. Okay, so that was our first question. You can see, as is displayed, so this is all the phosphorylation data, but as is displayed in green, these are the sites that either positively or negatively, at a statistically significant level, correlate with glucose uptake. And when Elise ran those correlated sites through a pathway algorithm, we were gratified to see that a number of highly significant pathways were pathways that sort of seemed to make sense. You know, insulin signaling, glucose metabolism, glycogen biosynthesis, and so on. So that was rather gratifying. So can we then start to sort of use these data to tell stories about what's going on in muscle with exercise? So I just wanted to share with you now a couple of stories. There are many, but I've only got time for a couple. So one of the stories, I have to just give you this little background about GLUT4 trafficking. So as many of you know, we know a lot about vesicle transport in eukaryotes. And it's the docking and fusion reaction that encodes the ability of one membrane, in this case a GLUT4 intracellular storage vesicle, to dock and fuse with the plasma membrane, is regulated by these so-called snare proteins. And there is typically one snare called a V snare in the donor vesicle, or in this case, the GLUT4 vesicle, that forms an extremely high affinity interaction with two other proteins, so-called T snares, found in the acceptor membrane, in this case, the plasma membrane. Now, as is the case in synaptic vesicles in neurons, for GLUT4 vesicles, the V snare is a protein called VAMP2. Now, the reason I bring that up is that one of the proteins that Elise identified on the list of those who exhibited a phosphorylation site that was correlated with glucose uptake was VAMP2. So here you see, this is the variance in phosphorylation of VAMP2 at this particular site, plotted against glucose uptake. And, you know, this is really quite an impressive correlation. I mean, R equals 0.85. For data of this kind, I must say I was quite struck by that. Now, we're in the process now of trying to functionally analyse the significance of this. But intriguingly, that phosphorylation site is located right here in VAMP2, which is at a position in the helical domain in VAMP2 that forms this high affinity interaction with the two T-snare proteins, in this case, syntaxin 4 and SNAP23. And what's really fascinating is that there is an amino acid at this position in the T-snare that is a positively charged residue, an arginine. And we're excited by the possibility that if you were to add a phosphate at this position in VAMP2, this may significantly enhance the affinity of this interaction. So this could be one of the players that is regulating the potentiation of insulin action in the post-exercise state. Now, if we next ask, well, if we focus on these positively correlated sites, what are the predicted kinases that are regulating their phosphorylation? And here is a list of kinases that Elise identified. And as you can see, the number one predicted kinase was mTORC1. And I don't think this kinase needs any introduction. It's one of the most researched kinases in the literature, plays a key role in nutrient sensing and many other facets of biology. Now, mTORC1 is downstream of AKT in a signaling pathway. So you might ask, well, is the upregulation of mTORC1 simply a function of there being an upregulation in AKT? And in fact, that is not the case. So here you can see that we were able to observe AKT phosphorylation at its two major regulatory sites, threonine 308 and serine 473 in these data. And you can see that both of those sites are increased with insulin, but not in a way that is potentiated with exercise in any of these subjects. And GSK3 is an AKT substrate, and that would be consistent with this observation. So what we have here is active or potentiation of mTORC1 kinase activity in a manner that does not correlate with one of its major upstream kinases. Now, of course, other things can activate mTORC1, and that's, of course, now an area of great interest for us, that there may be an exercise route into mTORC1 independently of the AKT pathway that is encoding this potentiation. Okay, so what Elise next did was asked, if we take these sites and ask which sites are correlated most with the other sites in all of these data, because that sort of, again, gives you an indication of which one's the most interesting. You can see here that there is a sort of a triangular zone of very highly correlated sites. And within that zone, there are some known substrates of mTORC1, as well as S6 kinase, which is a downstream kinase that's activated by mTORC1. So if we zoom into that triangle, here are the sites in this triangle. And what's fascinating is when Elise focused in on the amino acid sequences around these phosphorylation sites, you can see that many of them are predicted mTORC1 sites, and a couple of them are actually S6 kinase sites. So these guys are very much involved in the mTORC1 kinase network. So, which is activated and potentiated post-exercise. Now, here's one that caught our attention. This happens to be the alpha-2 subunit of AMP kinase, okay? Now, AMP kinase is known to be regulated by mTORC1, but in a negative regulatory manner. And to our knowledge, this particular site, serine 377, has not previously been described, certainly functional. So it was a fairly good mTORC1 consensus site, and we saw this, as is displayed here, like VAMP2, as being a site that is highly correlated with glucose uptake, R value 0.75, okay? And here's the actual data for AMPK alpha-2 377 phosphorylation. So you can see that it's hyperphosphorylated in the exercise leg after exercise compared to the rested one, and it undergoes a further potentiation upon insulin infusion. Okay, so very interesting. So then what we've since done is we've started to interrogate this because we got quite excited about it. And you can see in HEX cells, we've been able to observe also phosphorylation of this site at 377 in the alpha-2 subunit. And when we add either rapamycin or another mTORC1 specific inhibitor, phosphorylation at this site is almost completely inhibited. And in turn, on the other side of this slide, you can see that this is an in vitro kinase assay where we incubate the alpha-2 subunit with mTORC1 in vitro. And this is a very, very robust phosphorylation at the 377 site that we see when mTORC1 is added to the reaction. Now, as I said, typically we think of, when we think of the crosstalk between the mTOR pathway and AMPK, we think of mutual exclusivity. AMPK is a stress sensor. So AMPK is typically activated when things aren't going so well, starvation or other stresses. Whereas mTORC is generally activated when the system is ready to proliferate or synthesize protein. And so oftentimes when one is on, the other is off, okay? And indeed, there are sites, AMPK sites, on mTORC1 whereby AMPK inhibits mTORC1 activity. So what we're suggesting here is a situation where after exercise, a very unusual situation where we still have the sort of residual stress effects of exercise, so AMPK is still activated, but now in the post-exercise state, mTORC1 is also activated. And in fact, this is probably why one of the beneficial effects of exercise to fight cachexia and so on, people know that if gym junkies know about this, you go into the gym, you exercise, and then you have to take a protein shake immediately after your bout of exercise. And that's because probably mTORC1 is activated for several hours after you exercise. Now here, we're talking about something different. So we want to know, what is the impact of mTORC1 phosphorylation on AMPK? Is it blocking AMPK or is it activating AMPK? So in collaboration with our colleague in Melbourne, John Oakhill, he has cells that are deleted for AMPK. You can then re-express either wild-type AMPK in grey or an AMPK alpha subunit, which is mutated at this 377 site, okay? And surprisingly, what we've observed is that when we mutate this site, this results in a diminution in AMPK activity towards these two substrates, acyl-CoA carboxylase and ULK1. So this is consistent with the fact that when AMPK, at least the alpha-2 subunit, is phosphorylated at this 377 site, this activates kinase activity. Now, also surprising is that we do not see this behavior for all substrates. Okay, so here are two other bona fide AMPK substrates, and they appear not to be regulated in a 377 dependent manner. So this is very exciting, and we believe that, again, this is underpinning potentially another very important aspect of exercise biology, whereby AMPK, and so mTOR is activated after exercise, and again, we believe this is in an AKT independent manner. I think it's going to be very exciting to pinpoint the mechanism by which mTOR is activated by exercise. And when activated, it then phosphorylates AMPK on this alpha-2 subunit, okay? And we think this is very important. So you do not observe this phosphorylation on other subunits of AMPK, alpha-1 and so on. And then when AMPK is phosphorylated on that side, it somehow leads to the selective activation of AMPK towards some substrates, such as ACC and ULK1, but not other substrates. And again, I think we don't yet understand how that happens yet, but again, a very interesting and important thing to study. So what I've hopefully convinced you of this morning is that studying protein phosphorylation, I think, gives us a very unique and powerful window into biological function and phenotype. Moreover, and we're very excited about this concept of using individual variants in biological responses between people, something I think that many of us have ignored for a long time, but actually we think not only is it meaningful, but you can utilize it to filter out noise and get to the business end of phenotypes of interest. And that's, I think, hopefully you're convinced is being illustrated with that last story that I discussed with you this morning. So I thank you for your attention. I hope, you know, all of you have been able to have a coffee or whatever whilst this has been going on and are ready to go off and have a dozen Zoom calls or whatever it is that is absorbing you at this time. I think I've acknowledged most of the individuals that have helped with this work, and thank you again for your attention. Richard.
Great, thank you, David. That was awesome. I've got 3 pages worth of questions to ask and only 4 minutes in which to do it. So I may come back to you with some of my questions another time if I get the chance to. That'd be great. But a few questions for you. One has come in right at the beginning. It says, what is David's view on the barrier to identifying other kinases out there out of the 27,000 uncharacterized activated by exercise? Before you answer, we're about to experience what lockdown really is with my dog, wife and child about to run back into the house. So I'm going to go on mute just so you don't get any of that.
Okay. Look, this question, you know, I could run on all night. It's a great challenge to do this. And I think I presented for you there at the end what the challenge is all about. You know, just to interrogate that one site on AMPK at 377, it requires a lot of effort. You know, we were fortunate, we found a colleague who had knockout cells. We could then express back in the wild type and the mutant. But, you know, maybe we're just not smart enough to figure it out. But still, I think we're at a point where this remains the level of interrogation that is required to interrogate the function of all these sites. And it's hard work. And, you know, I hate to say that for many, many sites, you come up empty. We don't know why that is. Some would argue it's because some sites are not functional. I'm not yet convinced that's true. What we often find, for example, is that very important regulatory proteins, for example, TBC1D4, TSC2, you know, proteins of this kind, are not just phosphorylated on one site. They're often phosphorylated on multiple sites. And there's a lot of evidence emerging now, both from our lab and others, that when you mutate just one site, that might not have much of a functional outcome. And that's because the function is encoded by the multiple site phosphorylation. And we're finding that if you want to really get functional outcomes, you might often have to mutate 4, 5, 6 sites in the one protein. Again, really challenging stuff. But if you want to get to the bottom of it, we think that's what has to be done.
Great, thank you for that, David. I just got time for one other quick question. It's from Laura. I think she's referring to the single-legged exercise, and she asked, did you have any idea of the habitual exercise habits, in healthy individuals prior to the experiment? I think yes, that's what she's asking.
Yeah, look, we often get this question, and you know, it's a fantastic question. You know, so our Eric and Jörn in Copenhagen, of course, are responsible for all this, and they always tell us the studies were done in untrained healthy Danes. Now, for those of us who've been to Copenhagen, you know that an untrained healthy Dane is somebody who's probably riding about 20 kilometers on a bicycle every day. So, look, I don't think they're super athletes, any of them, but they're just healthy individuals, but I imagine, you know, in reasonable shape.
Thank you, David. And a quick question, I'm only going to ask this one because it's partly what I wanted to ask, is do you know which kinase phosphorylates VAMP2?
No. So one of the predicted kinases is kinase. And in fact, there is an old paper from many years ago by Richard Scheller's group describing that site as a kinase site. There was no functional analysis of it. That was all work done in neurons. So, but again, we'd have to go through all of that belabored work that I described to really prove that it was a kinase site.
And what about the STEM1 protein you mentioned right at the beginning? Obviously, there's at least a rationale for that to be involved somewhere along the lines of, you mentioned CAMK.
So no, that's an AMPK site. So that's actually described in the EMBO journal paper, if you're interested in following up on that. In fact, it's phosphorylated at 2 separate sites by AMPK. It is also phosphorylated by cyclic A kinase. But our study focused in on the AMPK site regulation. And, you know, I probably don't have time to belabor all the detail. I mean, it's a fascinating protein. It sits in the ER, and it has a conformation where it has a closed conformation in the absence of calcium. And then when calcium comes in, this protein flops open into an open conformation and now forms an interaction with a cell surface channel protein called ORI1. And this AMPK phosphorylation site sits right in this junction at this in this conformational complex, and that's one of the reasons why we got so excited about it. So
I can imagine.
Again, you know, I think often when before you leap in to study a candidate, you look for things like that give you a hint about, oh, maybe that's worth all my efforts, you know.
Yeah. Yeah, sorry, David, I could take up the rest of another hour with questions, but I won't. My main reason my wife would probably kill me. So I think all that's left for me to do is just offer you our, you know, sincere thanks for your time and your insightful talk. You know, truly groundbreaking stuff in science, and got remarkable potential for future candidates, as you mentioned before. So thank you, and I'm sure everyone will come with me and say thank you for your time. Just before I sign off, just a quick reminder or an update that we do have another talk on the 6th of May. I think it's 5 p.m. UK time by Stu Phillips, Professor Stu Phillips from Canada. If you want to know any more details, then just drop me a line, an email. It's Richard Mackenzie at University of Roehampton. David, I'll be in touch very soon to discuss some things we've been